started. First is the content. Content, introduction, discovery, genetic code table, salient feature. Next, introduction. The pathway of protein synthesis is called translation because the language of nucleotide sequence on mRNA is translated into the language of an amino acid sequence. The process of translation requires a genetic code through which the information contained in nucleic acid sequence is expressed to produce a specific sequence of amino acids. Next is the introduction of genetic code. The letter A, G, T, C means adenine, guanine, thymine and cytosine corresponds to nucleotide found in DNA. They are organized into codon. The collection of codon is called genetic code. For 20 amino acids, there should be 20 codons. Each codon should have 3 nucleotides. Impart specificity to each of the amino acid for a specific codon. 1 nucleotide, 4 combinations. 2 nucleotide, 16 combinations. 3 nucleotides, 64 combination, most suited for 20 amino acid. Here is the discovery of amino acid. Crick, Brenner et al. Experiment demonstrate that codons consist of 3 DNA bases. Marshall, Nirenberg and Hendrik J. Marthy in 1961 reported the first 3-letter code word for each amino acid. Hargobind Khurana, Genetic Code Dictionary and preview the results confirmed. Hargobind Khurana is an Indian scientist which give genetic code dictionary. Here is genetic code. What is genetic code? So, genetic code is a dictionary that corresponds with sequence to nucleotide and sequence of amino acids. Words in dictionary are in the form of codons, means A, adenine, G, guanine, T, thymine and C, cytosine. Each codons is triplet in nucleotides, means 3 codes are present. 64 codons in total. 3 out of these are non-codons. Non-codons means not take part in the genetic code. 61 codons for 20 amino acids. Here is the genetic code. Please see properly this genetic code table. Here is first letter, second letter and third letter. First letter is U, C, A, G. Second letter is U, C, A, G and third letter is also U, C, A, G. But in third letter, take 4 parts. U, C, A, G, U, C, A, G, U, C, A, G and U, C, A, G. Take 4 part here. And as it add, first letter, first box, U, U, U. Here is the phenylaline. And as these stop codons here is present 3. That is UAA, UAG and UGA. Next, you can take a screenshot of this. Salient feature. Salient feature is number 1, triple codons. Number 2, non-overlapping. Number 3, non-punctuated. Degenerate. Number 5th, unambiguous. Number 6th, universal. Number 7th, wobbling phenomenon. Number 8, terminator codon. And number 9th, initiator codon. We understand one by one. First is the triple codon. What is triple codon? Then, consecutive sequence of 3 bases on mRNA is called triple codon. That is, for example, UUU, that is phenylalanine. Here is tRNA diagram, and it is the triple codon. Next, genetic code, non-overlapping and non-punctuated. All codons are independent sets of 3 bases. There is no overlapping. Codons is read of a fixed starting point as a continuous sequence of bases taken 3 at a time. The starting point is extremely important, and this is called reading frame. Here is non-overlapping. Non-overlapping means, here is given an example to show the non-overlapping example. Non-overlapping example is the continuous series of genetic codes. Non-punctuated. Non-punctuated, consecutive or continuous. For example, non-punctuated means non-consecutive and continuous. Continuous means it takes continuous flow of genetic code, not oblique or any punctuation mark. Here is the genetic code redundant. Redundant genetic code is also called degenerated. Although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons except tryptophan and methionine. Each amino acid has multiple codons. Here is again shown the genetic code. Next, degenerate. Here is the degenerate or reductant. Some degenerate or reductant amino acid are given below. Next is the genetic code specificity. Specificity genetic code is specific or unambiguous. A specific codon always codes for same amino acid. Example, UUU here codes for phenylalanine. It can not code for any other amino acid. Only it codes for phenylalanine. Next, unambiguous specific. A particular codon always codes for same amino acid. That is, for example, UGG is tryptophan. Next, genetic code, universal. Universal, in all living organisms, genetic code is the same. The exception to universality is found in mitochondrial codon, where AUA codes for methionine and UGA for tryptophan instead of isoleucine and termination codon respectively of cytoplasmic protein synthesizing machinery. AGA and AGG codes for arginine and cytoplasm, but in mitochondria they are termination codon. Next is the wobbling phenomenon. Wobbling phenomenon is said that the rules of base pairing are relaxed at the third position so that a base can pair with more than one complementary base. Some tRNA anticodon have inosine at the third position. Inosine can pair with U, C or A. This means that we don't need 61 different tRNA molecules. Only half as many are required. In this codon and anticodon defined. Next is the nonsense codon. Nonsense codon is there are 3 codons out of 64 genetic code which do not encode the amino acid. These are called termination codons or stop codons or nonsense codons. There are 3 names of nonsense codons. That is one of the nonsense codon, termination codon and a stop codon. The stop codon are UAA, UAG and UGA. They encode any amino acid. The ribosome pauses and falls off the mRNA. Next is the terminator codon. Terminator codon is umber means UAA, ochre UAG or opal codon UGA. Here is the example. You can see and write down in your notebook. Here is the initiator codon. Initiator codon is AUG is the initiator codon in majority of proteins. In a few cases, GUG may be the initiator codon. Methionine is the only amino acid specified by just one codon AUG. Thank you.